What Jean asked me to do was give a brief overview to how we can use genetics and think about the application of genetics within a captive situation. So what I'd like to do today is give a brief overview of what genetics is all about, introduce you to some of the terms that you might hear floating about, and essentially tell you why this helical molecule, DNA, is really important for our understanding of what's going on both in captivity and in the wild. So after yesterday, it's no surprise that the world is in the grips of a major biodiversity crisis. This is something that we see happening in both terrestrial and marine and freshwater environments, where species are going extinct at a level about, estimates range from about 100 to 1,000 times elevated from the normal baseline levels of extinction. So this is putting increasing pressure on animals, and it's putting a focus on the need to have fallback plans to understand how this extinction risk and this biodiversity crisis is influencing the genetics of these animals. So I'm going to focus specifically on elasmobranchs. We had a really good overview of this yesterday about some of the threats facing elasmobranchs. So fishing, finning, and habitat destruction or degradation. These are all things that Ali dealt with in her talk yesterday, so I won't go into them in detail. But what I will say is that while I am focusing on elasmobranchs today, the basic principles that I'm going to be talking about can be applied to any animal, and any animal that is something that's of interest, and it's something that can be expanded very generally. So I think to start off with, we can think about genetics is why should we bother to care about this? I mean, there's a lot of expertise in this room about doing fantastic work, so why should you have yet another thing to think about? Well, genetics, I think what the best reason to focus on genetics is what it's all about. And essentially what it is it's telling us about inheritance. So if we think about a very simple family of sharks where we have a male and a female together who produce a bunch of offspring, what we do know generally is that the genetic material to produce these offspring, half comes from the sperm from the male, and the other half comes from the eggs from the female. So if we color code these into a male and female color, essentially what we're looking at is the offspring have half the genetic material of both the males and the females. And so that basic idea of inheritance can tell us quite a lot about what's going on within an animal. And usually we think about this down here, thinking about how that happened to get to this point where you have a genetic makeup of this animal, but we can expand this simple pedigree much farther back in time, even if we don't know anything about that pedigree, just by knowing about some of the laws of inheritance and how this genetic information is transferred down generations. And that's the kind of thing that I'm going to be talking about today. Before I do, I'm just going to give a very, very brief refresher in genetics, or maybe an introduction if you haven't come from a biological background. Now, this is a DNA molecule. They're these helical molecules that come together, and they look very complicated and fancy, but really what we can do is focus in on just part of this. And within this molecule, there are two strands that form the backbone of this helix, and coming off of those strands are these things called base pairs. There's only 4 of them, and they form combinations together. A's go with T's, C's go with G's. Now, really what we're after when we're thinking about genetics is trying to understand the order of these base pairs together. And we call that the sequence of those base pairs. And why we're interested in understanding that is that if you manipulate those sequences, or if those sequences change in any ways, it affects what DNA does. And the main thing that DNA does is it makes proteins and functional units that are really what keeps life going. So the order of these base pairs on a strand of DNA is what shapes the way these proteins act, and that's what maintains life essentially. Now, there are big parts of the DNA, or some parts of the DNA that produce things like proteins or functional units. This is called coding parts of the DNA. But the majority of the DNA actually doesn't do anything like that. It's non-coding DNA. It doesn't do anything useful as far as we can tell. And instead, it produces sections of it that are repeat units called microsatellites. And these repeat units are really useful to screen a population very quickly to figure out what's going on across a genome in regions that are accumulating differences over time. Now, this is something that I think Jean will tell you a little bit more about in his talk in a few minutes, but I just wanted to introduce the concept here. So we have things that code for something that does something in the body and things that just hang around. Now, why do we when we're thinking about these coding and non-coding parts, they really basically do the same thing over time when we think about it. So if we think about a population that has DNA, over time that DNA changes. And I think an important thing to get across in our minds conceptually is that DNA is constantly changing over time. Now the way I like to think about it is if you have some coins in your pocket or your purse. If you have some coins on a Monday, chances are by Friday you'll still have coins there, but they're going to be a different combination of coins. You might have bought a coffee, you might have paid for a bus ticket. Those coins are staying still, but some are coming and going. You're getting different ones. Now, if you like to do some international traveling, perhaps the Maldives, or maybe somewhere in Europe, you could have a situation where the coins in your pocket at the beginning of your trip, if you stayed in the UK, you'd still have British coins. But if you moved to the continent, you'd start to get euros in your pocket. And so you get a gradual change in your sequences that happen over time. And by knowing that this gradual change is something that is an inevitability of life, we can start to understand how populations are connected and how these, which populations have seeded, say, different parts of your collections. So understanding and incorporating this idea of change through time is really an important part of understanding why it's important to incorporate genes into captive breeding programs. But specifically, when we're thinking about genetics, genetics typically focuses on specific genes. Now, you have a large stretch of your DNA that gets packed up into chromosomes, but if you unpack that, a small section of that is the active part that you might be interested in, and that's called a gene. So genetics focuses on genes, but increasingly you'll hear the term genomics. And genomics focuses on all the hereditary information that's around in your cells, so essentially all of your DNA. And so genomics focuses on the whole genome. So we have two different levels of analysis and two different levels of focus. And these are there's sort of an interplay between the two of these, but these are things that are more and more becoming an important part of keeping in mind. What's happening at the level of the gene and what's happening at the level of the overall, all of the genes put together in all of your DNA. Now, I told you about the thing that we're really interested in is trying to understand these sequences of base pairs and how they're put together in the genome. So not surprisingly, if we're trying to understand what's going on in a gene, we use techniques called sequencing. And this is something where we're just trying to figure out what is the combination of these base pairs in a row. With genomics, we use something called next generation sequencing. So scientists aren't very advanced, so we had originally sequencing and then something else came along, they called it the next generation sequencing. What the nice part about next generation sequencing is it allows us to do things at an incredibly rapid pace. So using traditional sequencing methods, for example, the human genome, it took about 13 years to sequence the entire human genome. With next generation sequencing, you can do that in an afternoon now. So it's an incredibly rapid advancement in how we can use these techniques. And what I think is really nice, particularly from the point of view of sea life, is that we can apply these to animals that we don't really know a lot about. So using a combination of next generation sequencing and sequencing approaches with some work with ungulate rays, we were able to take an animal that we didn't know anything about and get workable genetic markers relatively quickly. So what I guess I'm trying to get across is don't be shy about what animal you have and whether or not there's enough information available about it existing today, because that's something that we can rapidly put together. So what I'd like to advocate today is that one of the nice things to do with genetics is link this idea of what's going on at the genetic level with captive management of animals, and in effect use this to create happy animals. And so what do I mean by happy? It's specifically a genetically diverse population, a genetically diverse population that maintains the amount of diversity that's present in the wild population, that avoids hybridizations amongst animals and avoiding that by knowing who's who, and prevents or minimizes inbreeding. So all of those things, I think, are what genetics can offer directly to captive breeding programs to try to maintain a healthy and viable population. So when we think about the current state of affairs of captive breeding programs, this is from a review a few years ago, but if we look at the taxonomic distribution, almost half of all captive breeding programs currently focus on ungulates and carnivores. Next, we go to birds, then a smaller subset of primates and reptiles and turtles. Small mammals are next, and finally we get to fish. Now, fish, and particularly elasmobranchs, are very understudied or very underrepresented in breeding programs. What's been really nice over the last 2 years is seeing the growth of captive breeding programs amongst elasmobranchs with the development of stud books and starting to get a lot of the foundation in place. So I think if this chart were to be updated, this line or this end part of the graph would already be moving up. And that's thanks in large part to the efforts of people here in this room. So with this background in conservation and knowing that there's some work to be done as far as linking conservation breeding programs and genetics in fish and elasmobranchs in particular, I think what we can address are a few key issues. So what genetics I think can help us do generally, I'm going to break them up into 4 topics. 1, answering this question: who are you? Where did you come from? Who's your daddy or mommy? And are you in trouble? And it's those 4 that I'm going to be talking about as ways that are potentially something that you could apply in your collections. Now this isn't an exhaustive list, it's just a start. So who are you? Using genetic techniques, we can identify which species are which, and identify species based on what's called DNA barcodes. This is small parts of the DNA that all animals share, which you can very easily look at to determine if you're the same species or not. So it sounds very simple, and technically it is quite straightforward to do, but what it means is that you can take 2 species that look morphologically very, very similar, like these different species of cow nose rays, stamp a DNA barcode on them, and identify which species are which. The nice thing is this is a technique that you can apply within collections. It's also a technique you can apply in the wild to sort out which species that you think may or may not be the same, and to identify cryptic species. Often, particularly for the elasmobranchs, species look very, very similar, and teasing them apart and figuring out who's who can be difficult. And this is a way that you can rapidly do that. Next, where did you come from? So because DNA changes slowly over time, it leaves a signature. And so we can look at different populations and identify where they've come from based on these genetic signatures, which tells you something about the origins of a species and the geographical origins of a species. So here's a recent example of a spot-tail shark in Southeast Asia and Australia. And in each of these different locations, the colors, the degree of similarity in the colors tells you about how closely genetically related those species or those individuals are to one another. So within these species, Australian populations tend to be much more closely related than they are to those around Indonesia, whereas the Indonesian populations tend to be more closely related to one another. So essentially, we're starting to see geographical isolation and geographical variation in the genetic signatures among populations. Now, this can have some implications for management because individuals tend to become very locally adapted to their conditions. So the way that your genes and your DNA are adapted to your local conditions may differ between populations, which means maintaining populations in captivity. If you mix these up, individuals from different populations, you can start breaking down these adapted gene complexes. And so it's something that's worth keeping in mind and considering when planning breeding in captivity. So next, who's your daddy or mummy? So it's a very clear and obvious thing that often animals are kept together in mixed sex aquaria display. And when you have a situation like this, it can be very difficult sometimes to figure out who's been breeding and which offspring are being produced. So because we know about this genetic inheritance, we can very quickly and easily now, for many species, identify if you have a mixed group of individuals and you have an offspring that's produced and you want to find out where that offspring came from, you could very quickly say this individual shares the genes of these 2 individuals, making those individuals its parent. So knowing where an animal comes from means that you can start to keep track within a mixed sexed tank. You can start keeping track of who's breeding. You can also start to keep track of how you want to pair this animal in the future. Finally, are you in trouble? So this is the bit that I think is where genetics offers the most practical application for captive breeding. And it's using genetic tools to assess the genetic diversity within a population. Now, genetic diversity tells you how different an individual's genes are. And what this is really important is because it's linked to disease resistance, it's linked to viability of offspring, survival rates of offspring, and usually animals can do pretty well until a challenge comes. And when you have a challenge, say a rapid change in temperature, change in salinity, anything like that that might, or the introduction of a pathogen or a parasite, that's when animals that have a higher level of genetic diversity tend to do better and survive better. So within a captive situation, we can think about trying to maintain this diversity by looking at levels of inbreeding. So if we think about a parent and offspring, just using a very simple graphical display, an offspring, or if the parents are outbred, it means they typically have different copies of their genes from one another. So these individuals are both different from one another, but they both have different copies of these genes. They will then pass one of these on to the offspring, and in an outbred situation, chances are the offspring will then get 2 different copies of each gene, meaning they have high levels of genetic diversity. And that's really a safeguard to prevent against all of life's problems that get thrown at all of us throughout our day-to-day life. If you're looking at situations where animals are inbred, it's more likely that the parents are going to have similar copies of genes. So when these individuals breed, the offspring then get produced with the same copies of genes, which means they have less scope to respond to any of the challenges of life. And so this is basically why it's important to have more outbred individuals than it is to have inbred individuals in your population. When we think about that from a management perspective, looking at a captive population, if you have a pedigree, for example, of some wild-caught animals and some captive-bred animals, which each of the colors in this example representing a different level of genetic diversity, we can ask a couple of questions with this by knowing how individuals are related, or how individuals are genetically diverse. We can ask, was the starting population sufficiently genetically diverse that we can have a healthy population? So we can look at this starting population and say, were the individuals that were brought in from the wild genetically diverse, or did we happen to collect individuals that were already so far inbred that it's going to be really challenging to make a viable and sustainable population going forward? Within the captive-bred individuals, we can ask, are these captive-bred individuals inbred currently? And are there ways that you can pair individuals up to minimize the chance of them being inbred? And we can ask, are captive individuals that are maintained over time in a population maintaining the level of genetic diversity as their wild ancestors? And maintaining the levels of genetic diversity is a really important thing because if there's ever any links back to the wild, it's having a population in a captive situation that reflects what's going on in the wild is a very useful thing to have. So putting this all together, so the way I view genetics and the way I view genetics in a conservation setting is that aquaria have a very important part to play in understanding, developing and understanding the importance of genetics as far as maintaining population health. That's not something, though, that can be done in isolation. And I think clarifying the links with wild populations is an essential part. So there's this constant back and forth between what's going on in captivity and what's going on in the wild. And linking this with the lab tools to actually identify the genetic aspects of these breeding programs, and then getting a nice circle between here where there's lab integration, looking at what's going on in the wild, linking that back with what's going on in captivity, and back and forth. Now, this is a model that's very easy to put onto a PowerPoint slide, but it's very difficult to do in reality. One of the nice things about the last 2 years is that we've been doing this a little bit, or getting this off the ground with some work that Jean has been spearheading with the ungulate rays, where we've started to look at what's going on within the captive population and have plans to move forward to talk about what's going on in the wild situation and then link these two together. And I think overall there's a very tremendous capacity to link genetic information with understanding animal health and animal welfare and trying to understand the processes that are going on at the genetic level and how that can feed back into understanding what's happening both in wild populations and captive populations. So with that, I'd just like to thank you for your time. Our team at Manchester is Richard Preciosi and Jenny Roundtree, who were here yesterday but unfortunately had to leave. Graham Fox is a PhD student. Yulia Durolti was a project student who collected most of the ray data that Jean will tell you, I think, a little bit about more later. Sam Hook and Katie Hunter are here in the back, which a very nice thing is that Sam has got a NERC case partnership with Sea Life, where we can start looking at this in a more linked way. And finally, I'd just like to thank Emma, Jean, and Chris for all the support for the last couple of years of actually getting this off the ground, because I think it's been a lot of fun to do. Thank you.